Now we are seeing here regarding ethylene, an important plant growth hormone, and coming to some of the historical aspects with regarding to the discovery of this hormone. It was discovered in 19th century, and you can see that when coal gas was generally burnt, that is generally was used in the illumination of street lamps. The earlier street lamps was in such a design that buckets of coal were hung on big poles, and that was lighted up during the night, and coal gas was emanating from there. And in the morning they could see that a lot of leaves were falling down because of the effect of this coal gas. So this is one of the very early observations what they made with regarding to the ethylene. Now in 1901, Dmitri, who was a graduate student at St. Petersburg, he found that ethylene could bring about a triple response in pea seedlings, that is reduced stem elongation, increased lateral growth, sideways lateral growth, and abnormal horizontal growth. So these were the three important changes which he could observe in pea seedlings as a result of the ethylene response. Again, coming to the year of 1910, cousins, they invented or they found that oranges, when kept along with bananas, prematuring ripening of bananas took place. That means something was emanating from oranges which could bring about the ripening phenomena in the bananas. So these were some of the earlier observations which they made with regarding to the ethylene, and they could not really find out what was responsible, which chemical is involved in this particular process and all. But later on discoveries have made known that ethylene was responsible for bringing about these changes. And if you check the biosynthetic regions in a plant where the ethylene is being produced, you can see that meristematic and nodal regions are most active for the synthesis of ethylene. And also ethylene is synthesized in plenty during fruit ripening, during leaf senescence, during the abscission of the leaves, also during wounding stresses. If you make a wound on the plant, you can see that ethylene is being produced over there. And you can see that ethylene is very lighter than air, and it is inflammable, and ethylene gets converted into ethylene oxide, and it can get converted into ethylene glycol, or it can get converted into oxalic acid, and further it gets converted into carbon dioxide. So ethylene is a gas basically. So once it is there in the environment, it gets converted into these forms. And ethylene, if you want to trap ethylene, the best chemical solution for that is potassium permanganate, KMnO4, and this can trap the ethylene and you can use it for later use. Coming to the biosynthesis of ethylene, you can see that methionine is the precursor of synthesis of ethylene, and methionine gets converted into ethylene, and in between you can see methionine gets converted into adomet, that is S-adenosyl methionine, that further get converted into ACC, that is 1-aminocyclopropane 1 carboxylic acid. The conversion of adomet to ACC is facilitated by the enzyme ACC synthase, and this enzyme is regulated by various environmental and internal factors, and this enzyme is highly unstable and is purified using antisera. If you want to purify this enzyme, you can do it with the help of antisera, and later works have helped in the expression of this gene in E. coli and the synthesis of this enzyme in E. coli, and this enzyme showed similarity to aminotransferase superfamily of enzyme, and it is encoded by a multi-gene family. Once ACC is formed, ACC can get converted into ethylene, by the action of ACC oxidase. And so this is the biosynthesis process taking place in the plants. And as we have discussed in the earlier plant growth hormones, normally ethylene is found in conjugated forms. One of the prominent form is N-malonyl ACC, another one is GACC, and these two are the major conjugated forms of ethylene, and only ethylene will be formed as and when there is the requirement. Here I would also like to discuss about one of the phenomena called as ACC synthase gene, and we have already seen here this is the ACC synthase enzyme which is responsible for the conversion of adomet to ACC. The gene people have already worked out to develop antisense DNA for this gene. And what is the significance of the antisense DNA for this enzyme? Once there is an antisense DNA, no further development of for transcription process will take place, no mRNA synthesis will take place, no enzyme will be synthesized out of it. When there is no conversion of adomet to ACC because of the non-functional of this enzyme or non-production of this enzyme, that means no ethylene is synthesized. When no ACC, that means no ethylene will be synthesized. When there is no ethylene, that means one of the major physiological process controlled by ethylene will not take place, that is the ripening of fruits. So people have developed this antisense DNA in order to prevent the action of the ACC synthase enzyme, so that ethylene will not be produced, no formation of ethylene will take place, as a result of that no ripening will take place. One of the biggest advantage of this discovery is that you can have fruits in an unripened stage. The tomatoes which were synthesized earlier, these tomatoes were ripening very quickly before its transportation to its destination sites. And normally it takes 2 or 3 or 4 days or maybe a week's time for transportation of tomato from its site of cultivation to site of usage. By the time it reaches over to the place of its usage, the whole tomato is destroyed because of the over ripening. But with this invention, the antisense DNA technology in the case of ACC synthase gene, the prevention of ethylene, the formation of ethylene is prevented, as a result of which ethylene is not synthesized, no ripening takes place. And in this case, how then ripening can be achieved? Once the fruit is transported to the area of its usage, you apply ethylene over there artificially and you can ripen the fruit. So the ripening can be controlled as and when required by a person. So that is one of the biggest advantage of the antisense DNA technology developed in the case of ACC synthase gene. Ethylene production, as I told you, can control the fruit ripening. There you should remember about the ACC synthase and ACC oxidase. You can control these enzymes by antisense DNA technology, and you can control the ripening of the fruits. Also, ethylene production is stress induced. These sort of stresses like wounding, chilling, flooding, drought, all can enhance the production of ACC synthase. As a result of this, quick senescence of the plant will take place. And auxin can induce the ACC synthase gene. So ethylene production can be influenced by basically these 3 processes. The fruit ripening can influence, the stress such as wounding, chilling, flooding, drought can influence, and auxin also can influence. There are various inhibitors for ethylene production. One of the major inhibitor is AVG, that is amino ethoxyvinyl glycine. In short, we call it as AVG. Another one is AOA, that is amino-oxyacetic acid. This can inhibit the conversion of adomate to ACC and prevent the synthesis of ethylene. Another major inhibition can occur in the conversion of ACC to ethylene. So, this is the first step of the biosynthesis, adomate to ACC. Second step is the conversion of ACC to ethylene, that can be prevented by cobalt. Now, ethylene action can be inhibited by silver also, Ag plus silver ion of AgNO3. It can be prevented by carbon dioxide around 5 to 10%. So, this is the inhibition of ethylene action. Okay. So, here we have explained in the first two instances, we explained the inhibition of inhibitors in the biosynthetic pathway. In the second stage, we explained about ethylene action getting inhibited by Ag plus of AgNO3, that is silver nitrate, and carbon dioxide, that is around 5 to 10% of carbohydrate. Another inhibitor is trans-cyclooctene, which is a competitive inhibitor to the ethylene for binding to the receptor. So, this is another stage of inhibition, that is binding to the receptor. Instead of ethylene, trans-cyclooctene can come and bind to the receptor and thus prevent the ethylene to get inhibited, but no ethylene action will take place. So, ethylene inhibition can take place at the level of synthesis, it can take place at the level of ethylene action, it can take place at the level of the receptor. These are a list of various development and physiological effects. One is promotes fruit ripening. Second is leaf epinasty. When ACC from root is transported to the shoot, leaf epinasty, that is the bending of the leaf take place. Third, induces lateral expansion, that we have already seen the finding of Dimitri, which is the triple response of P. There we see that there is a lateral expansion, cell expansion takes place. Fourth, hooks of dark grown seedlings are always bent like this, that is because of the ethylene action. 5, breaks seed and bud dormancy in the case of cereals, peanuts, sprouting in potato tubers and other bulbs. 6th, promotes elongation of submerged aquatic species, because aquatic species, when they are submerged, when the level of water goes up, if the elongation does not take place, the plant will be submerged and will be destroyed, but ethylene will come into play and it will promote the elongation of the submerged aquatic species. So, the leaf and the shoot tip can always remain at the top of the water level. 7, induce root and root hairs. 8, induce flowering in pineapple. This is generally used for synchronization of the fruit set in pineapple, also for flowering initiation in the case of mango. Promotion of female flowers in cucumber, because only female flowers will bear fruit or bear the vegetable cucumber. So, normally for all these purposes, ethylene is being used. Again I repeat, for induce the flowering in pineapple, for promotion of female flowers in cucumber, flowering initiation in mango, synchronization of the fruit setting in the case of pineapple. It can enhance the leaf senescence rate. Senescence regulated by balance of ethylene and cytokinin. So, the leaf senescence can be, if you apply more of ethylene, the senescence of the leaves takes place. It can, ethylene can act in the abscission zone, is regulated by the auxin. This I will explain in one of the later slides. This is the triple response of etiolated pea seedlings, which Dimitri has found out. Seedlings show a radial swelling. This is the triple response. This is the normal pea seedlings, and this is the triple response: stunted growth, lateral expansion, and thickening. Horizontal growth of the epicotyle takes place here. Epinasty, this is already we found that because of the action of ethylene, in the list of earlier we found here the second point, leaf epinasty. one of the important physiological effects of ethylene. That is the downward bending of the leaves. Not here it is described in the case of tomato leaves, but it can happen in any leaves. Downward bending of the leaves takes place. Epinaceae results when the cells on the upper side of the petiole grows faster. Why? How does it bends down? When the upper part grows more, the leaf bends down. So epinaceae takes place because of the action of the ethylene. Here you can see inhibition of flower senescence by inhibition of ethylene action. Here this is the control. The flower is senescing very quickly. But here this is a case where this is being done in the case of carnation flowers. They were held in deionized water for 14 days in the left and right without the silver thiosulfate, a potent inhibitor of ethylene. So here we have a potent inhibitor of ethylene applied, that is STS, silver thiosulfate. It will inhibit the ethylene production here. Here there is no inhibition of ethylene production. So when there is ethylene production taking place, flower degradation takes place. But here the flowers are very live because the ethylene production has not occurred because of the inhibitor of ethylene production, that is silver thiosulfate. And as a result, the waste life of the flower is increased. This is having very important commercial application. If you want to increase the waste life of flowers, because you know some of the flowers like carnation in this is very expensive, and if you can increase the waste life from 1 week to 2 weeks, that means you are doubling the waste life, and that has got a lot of commercial application, and this way you can try to increase the waste life of the plants. Here you can see the promotion of root hair formation by ethylene in lettuce seedlings. Here a lot of here ethylene is applied towards the right, and here it is without ethylene, and a lot of root hair formation occurs as a result of ethylene application. Effect of ethylene on abscission in birch, that is Betole pendula. The plant on the left is the wild type, that is with ethylene formation. The plant on the right was transformed with a mutated version of the Arabidopsis ethylene receptor ETR1. So in the mutated case, no ethylene action will take place because the receptor is already mutated. And here this is a wild type, and because of the ethylene action you can see the whole leaves has fallen down, but here it is live. So again this has got a lot of commercial application. If you can increase the longevity of the leaves can photosynthesize for a longer period, and you can increase the productivity of the plants. Here I told you the ethylene can control the, we have already seen earlier, ethylene in abscission zone is regulated by auxin. I told you we will be explaining in a later slide, and here it is now. Normally there is an auxin gradient from the tip of the leaf towards the base of the leaf, and this will normally this occurs during the maintenance phase of the leaf. Leaf is live, green, photosynthesizing, but here you see when the senescence starts, shedding induction phase, it is called the shedding induction phase. At the base of the leaf, petiole region, more ethylene is produced, and the auxin gradient which we saw over here is cut out. No more auxin flow is maintained here because of the production of ethylene, and when there is no auxin gradient is produced or flow of oxygen from the tip of the leaf taking place, you can see that the senescence initiates and finally shedding phase occurs. That means the leaves shed from there because of the senescence happening. So this is the schematic view of the role of auxin in ethylene during leaf abscission. So the leaf maintenance phase occurs during the auxin gradient, but no ethylene. Induction starts when the production of ethylene starts, and finally auxin production is prevented or auxin flow is prevented, and in the shedding phase you can see there is the total senescence taking place. And now we come to the ethylene action, how it is occurring. Let me explain directly from the figure. Here we have got a protein. The RAN1 protein is required to assemble the copper cofactor into the ethylene receptor. Here we have the copper cofactor. The RAN1 protein is required. This is the RAN1 protein, and it has got a histidine domain, histidine kinase domain and receiver domain, similar to what we have found earlier in the case of cytokinin. Then ethylene comes and bind over here. Okay. So in the absence of ethylene, ETR1 and the other ethylene receptors activate the kinase activity of CTR1. See, activation takes place, ethylene binds, activation, the phosphorylation takes place. As a result of the phosphorylation of the histidine kinase domain and the receiver domain, deactivation of CTR1 RAF-like kinase occurs. And as a result of that, MAPkase gets activated. And as a result of that, you can see the inactivation of CTR1 allows the transmembrane protein EIN2 to become active. The inactivation of CTR1, the inactivation of the CTR1, activation and inactivation. So here the activation occurs. Once the inactivation occurs, what happens? The transmembrane protein EIN become active. Okay. And EIN1, 2 protein enters inside the nucleus. Activation of EIN2 turns on the EIN3 family of transcription factors. So EIN3 transcription factors are activated, and this transcription factors goes and bind to the gene responsible for ethylene synthesis. So this is the total activation process, gene activation process on ethylene binding. Ethylene bind to the receptor, the histidine kinase domain gets phosphorylated, receiver domain also gets phosphorylated, and CTR1 RAF-like kinase gets activated. And as a result of the inactivation of that, EIN2 protein get inactivated, and because of that, EIN3 gets activated and it moves into the nucleus, and that acts as a transcription factor for the ethylene responsive genes function. So this is how the gene activation takes place as a result of ethylene action. Coming to the commercial uses of ethylene, ethylene is difficult to apply in field because it is a gas. So normally ethylene releasing compounds are being available in the market. One such is called as ethophon. Ethophon is nothing but 2-chloroethyl phosphonic acid, and the trade name of the same is Ethylene. And you can buy it from the market in the form of this trade name Ethylene. And you can apply this ethyl into the field or into the plants and can bring about the ethylene action. And also I explained to you regarding one of the commercial applications is with regarding to the transgenics being developed with the antisense version of ACC synthase and ACC oxidase, wherein we can delay the ripening of the fruits, and you can later on bring about the ripening of the fruits as and when required after the transportation of the fruits to its destination. So with this we have come to the end of the session on hormones. So here we explained basically on 4 different hormones. One is on auxins, cytokinins, gibberellins, and finally on ethylene. We saw with regard to its some of the historical aspects with regarding to its discovery. We saw some part with related to its biosynthesis. We saw some aspects with related to its physiological functions, and finally we have seen regarding all these hormones how exactly the action is taking place on application of this hormone to the plants. So we have come to the end of this chapter. We will see another chapter on another session.